Hi everyone, my name is Lindsay and today we're going to be talking about epistasis. So first off, what is epistasis? Basically, it's a genetic phenomenon where a gene at one locus hides or masks the effect of a gene at another locus. These genes produce proteins that correspond to different versions of a phenotype. When epistasis occurs, the expression of one gene will stop the expression of another gene. This means that multiple genotypes are capable of producing the same phenotype. It's important to understand that epistasis involves two different genes. It is similar to dominance in that one can override the other, but dominance involves two different alleles at the same locus as opposed to two different genes at completely different loci. Now, for a simple Mendelian trait, we're used to getting the classic 9:3 to 3:1 ratio when doing a dihybrid cross. When epistasis is in play, this ratio begins to change depending on the type. There are 6 different types of epistasis, which I have listed here, and you can see that each type has a different phenotypic ratio associated with it. Today we're going to do an example with duplicate recessive epistasis. So with this type, the same phenotype is going to be produced when the organism is homozygous recessive for either gene. The other phenotype is only expressed when there is at least one dominant allele at each locus. To demonstrate this, we're going to be looking at coat color in the platypus. Okay, here's the pathway we would use to represent duplicate recessive epistasis. We start with the brown coat color being the beginning precursor that we start with. In order to move from the precursor to the next step, we must produce enzyme A. However, enzyme A can only be made when there is a big A allele or the dominant A allele. If a platypus has two little a's, it will not produce enzyme A and will express the brown coat color phenotype. If a platypus does have a big A, it will move on to the brown intermediate. While the brown intermediate is genetically different from the brown precursor, this type of epistasis causes both phenotypes to be the same. To reach the teal end product, that platypus with at least one big A will also need to produce enzyme B, which is only made when there is a big B allele. Thus, a platypus with a big A allele but two little b's will express the brown intermediate, and a platypus with a big A allele and at least one big B allele will express the teal end product. Let's look at a dihybrid cross and calculate the phenotypic ratios that would result. First, we need to write out the standard simple Mendelian ratios and then work from there. Using the pathway written above, we can determine which phenotype will be expressed for each genotype. For this cross, we can add up all of the brown individuals and conclude that the phenotypic ratio would be 9 teal platypi to 7 brown platypi. Next, we're going to do one more cross, but this time the parent will be big A, little b, and little a, big B, little b. Since this is not a dihybrid cross, I made a Punnett square for each locus and listed all the possible genotype combinations. Again, we will use the pathway above to assign each genotype to its expressed phenotype. Every genotype that is homozygous recessive at least one locus will be brown. Thus, the phenotypic ratio for this cross is 1 teal platypus to 3 brown platypi. Here's a list of my sources, and that concludes this video on epistasis. Thanks for watching.